Welcome back, everyone. I think we will get started. For those of you who haven't met me, I'm Matthew Gould, the Chief Executive of ZSL. And the first thing I wanted to say was just a huge thank you to you. Every day, I am acutely conscious that this is your society, that you are ZSL. And I wanted to say I really hope you are happy with your society. I hope you're happy with what we're doing, whether it's the Zootown project, which is giving a new life to the old reptile house, creating somewhere which I hope will be absolutely best in class conservation and nature themed educational play. Generations of 3 to 7 year olds going in, getting excited about nature. I hope you're happy about the fact that end of last year, we mounted an expedition to rescue 60 healthy Darwin's frogs when we thought that species was on the very brink of extinction. And they are now here breeding like crazy in a really gratifying way. I hope you're happy that literally, I think just through that wall, we have a new £2 million supercomputer that means that our scientists and conservationists can do really cutting edge work in AI and data, building on some of the brilliant things we've done to date. So I hope you're happy with what we're doing. I hope you're happy with where we're going. Our strategy refreshed strategy that we put out earlier this year. If you haven't seen it, please do. But it sets a very clear direction of travel, a very clear set of targets for us to try and hit. And let us know what you think. And I hope you're happy as fellows with how you're treated by your society. And we're always open to hearing from you. You've told us previously how you wanted this day to run better. But Pippa and I and the team will always be happy to hear from you, ideas, thoughts, suggestions, even complaints. It's my huge honor to introduce our keynote speaker today, Professor Anjali Goswami, who has, I'm not going to do the thing of trying to explain her science to you before she explains it to you much better than I will. But I will tell you just a little bit about her. She's a really impressive biologist at the Natural History Museum, a friend and longtime collaborator and partner with ZSL. She has just stepped down as the president of the Linnaean Society, where she was the first person of color to do that role since 1788. And I should say, not because in 1788 they had another person of color, but that was because the year it was founded. She, in 2024, became a fellow of the Royal Society. As you know, an extraordinary mark of esteem for her scientific prowess and leadership. And most importantly of all, in 2018, she got ZSL's scientific medal. She is about to become someone very important for ZSL, even more important than she is at the Natural History Museum. She is about to become the chief scientific advisor to DEFRA, where she will have an enormous influence and agency on the UK's environment policy at home and around the world. So we'll have a huge responsibility sat on her shoulders. But we are delighted that she's got this role. We are really looking forward to working with you. And if there were a pair of shoulders that was ready to take on that responsibility, it's yours. Thank you so much for coming. Your work is inspirational at the cutting edge of technology, conservation, and zoology. We're really excited to hear from you. Professor Goswami.
Well, thank you very much for that very kind introduction. I hope I live up to that. I'm very happy to be here speaking with all of you about my research, which will slightly take a back burner for the next few years. But this is a great opportunity to really revel in it for a while. So my my field is, I'm an evolutionary biologist. Originally working on mammals, but in recent years I've spread out across all vertebrates and even working on insects at times. And I take a paleobiological perspective, which means that, let's see if I can get this to run. I spend a lot of my time in places like this. This is one of my field sites in Argentina, where we're out looking for fossils from the period just before the last mass extinction, 66 million years ago. And this is a part of my research where we go out and try to really understand the factors that drive what species on Earth look like. And so I'm going to be talking about a lot of this over this next 45 minutes or so. And usually when you meet a paleobiologist, people assume that this was me when I was a child, right? Really, really excited about paleontology. And if not that, then it was this, right? But the fact is, I actually never even went to a natural history museum until I was at university. We didn't have one where I grew up, and I never even saw Jurassic Park until the pandemic when my kids wanted to see it. It's actually pretty good, but I really was proud of being the only paleontologist in the world who had not seen it. So this is where I grew up. I grew up in Detroit, Michigan, and you know, where you grow up has a huge impact on kind of how you feel about the world and your perspective on the world. So this partially shaped my perspective, but actually more importantly, when I was about 4 years old, my parents' visa extension was rejected, and we moved to India, which is where they're from. And so those are my parents there, looking very Indian on an elephant. But where my family is from is a state right in the middle of India called Madhya Pradesh. And Madhya Pradesh, has anybody ever been to Madhya Pradesh? No, a couple people. Okay. Well, MP is really famous for a number of things, but in particular, it's where the Jungle Book was set, and that's because it is a place with about half the world's population of tigers, of wild tigers. And so this is a picture that my husband took actually the last time we were there. So when I was 4 years old, living in India for about a year or so, I saw my first tiger in the wild with my cousins, and I remember it's my very first memory of life, actually, and it's really imprinted on me. And so, really, ever since then, I've been obsessed with tigers. I feel like I should have some more elusive species that I should be obsessed with. But I love tigers, but more importantly, it's really made me really obsessed with the natural world and with the immense diversity of species that we share this planet with. Now, of course, I have also actually written a book from my experiences there, but it's actually about carnivore evolution. So not quite as famous, but I think it has its moments. And really the perspective that my work takes is really trying to understand essentially why life on Earth looks the way that it does, why animal life in particular, although I dabble in other groups, why animals look the way that they do. And you know, the fancy scientific way of saying this is really what intrinsic and extrinsic factors shape the tempo and mode of evolution of organismal form. And I'm sorry, but this easy way of saying this is why do animals look the way that they do, really? And so these are the different factors that we consider. We look at things like development and genomics. We look at biomechanics and how animals move around. We look, of course, at their deep evolutionary history. We look at things like sensory evolution and brain size. And of course, we look at ecology and environment. We try to bring all these different factors together to really understand why why life on Earth looks the way that it does. Now, sometimes when you think about diversity, it's helpful to think about groups that aren't so diverse. And actually, not simply because this is really my 4-year-old self shaping my 50-year-old self's research program, but almost. But cats are actually a fantastic group to think about from the perspective of evolution because they are really not very diverse, actually. They are stunningly conservative in terms of their form and function. In fact, cats are the only groups of vertebrates that get above about 50 kilos in size and can still go into a crouch and move around in a crouch posture. You know, a lot of people have tried to figure out why we've had research grants really just focusing on like how can cats do what they do because they are incredibly impressive. You know, whether you go from the smallest species, just a couple of kilograms, up to the large ones that are obviously several hundred kilograms, they basically scale fairly consistently. And you know, so I think about this as a scientist, but occasionally also I think about this just as a person who has not very deep thoughts randomly on a Saturday morning. And I once wrote on Twitter a few years ago that part of this might be, we think about all the reasons, and I'll talk about them: developmental constraints, functional constraints, evolutionary history. But also, maybe they're perfect. Maybe they're just utterly optimized, this perfect, morphological and functional and niche, and they just don't need to be diverse. They just need to be perfect cats. Because I wrote this on Twitter, I immediately got piled on by everybody. If I'd put this on Blue Sky, obviously everybody would be like, "Oh, great idea," and then this would have ended at that point. But actually thinking about this, because I then had to argue back and forth with many people, I really started to think there was something in here about um, how we think about evolutionary success, how we think about diversification in different species, and how we think about adaptive optima. Um, and it got picked up by various things. So Scientific American did something on it. And actually, just a few weeks ago, randomly, if any of you listen to National Public Radio in the US, it was on Wait, Wait, Don't Tell Me as a question, quite randomly. So be careful what you tweet. I guess you guys all know this. But it is an interesting question. And actually, like I said, I think cats and also dogs are a really useful group, and domesticated species in particular, of course, as Darwin well knew, are interesting groups for thinking about evolution and why animals diversify the way that they do. And so if you think about cats and dogs, they have very, very different trajectories as domesticated species as well as wild species. And a lot of this is down to how they develop. So cats and dogs both start off in with these really round skulls. And in fact, you can think about it, most mammals do, fairly rounded skulls. And as they get older, dogs become much more elongate, at least in their wild forms, and cats a little bit, but not very much. So hopefully you can see that the cat, as it goes ontogeny being development, as it gets older, still pretty round. Dogs become far more long in their face. And this difference in developmental change is actually what gives breeders a lot to play around with dogs. And so when you can basically select along this developmental trajectory with dogs and create a lot of different morphologies or different shapes. Whereas with cats, there's just not that kind of variation. And that's why when you have, these short-faced cats, as this is my cat and dog. Well, my former cat and dog, they were my grad school pets, are long dead, but still my favorites. But as they grow up, you can see that Eli, the dog, got very snouty, and Chairman, the cat, less so. And we call what cats do isometry. So shape change through time, cats kind of stay the same. Whereas dogs have allometry, they get much longer, they have a lot of shape change through time. Now, as I said, allometry gives breeders and evolution a lot more variation to play with. And that's why you can have dogs that look like this. The dog in the middle is my current dog, Casper, with his glorious 13-centimeter long snout. And this abomination next to him is this extremely short-faced pug. Whereas if you really want to create a monstrous cat, the worst you can do with it is take its fur off. You really can't embarrass them in any other way in terms of domestication. So that's part of the story. There's also ecology plays a role, of course. And there's a strong argument that cats are less ecologically diverse and also, as a result, possibly less evolutionarily flexible. So if you see where the arrows are pointing, those are the carnassial teeth. These are the teeth that are for slicing meat. Now, hopefully you can see in the dogs, they still have teeth behind those carnassials. Those are their molars, and they can still grind up food with those. But with cats, they've actually lost everything past that slicing dentition, and that really locks them into this carnivorous lifestyle. Whereas dogs can still eat various other things like a head of broccoli. It's a lab, so obviously it would just eat anything anyway. And we can show this quantitatively that actually hypercarnivores like cats actually evolve slower and evolve less diversity in terms of their form compared to their less hypercarnivorous or hypo or mesocarnivorous close relatives. So things that are within the family Carnivora or the order Carnivora, but actually can still have a wide variety of diets. And so this is part of the reason why I think cats are an interesting group, because they are unusual in many ways. They're quite locked into this specific morphology for various reasons. But as I said, I think that's also, there's a positive side of thinking about this, is they're just very good at this. Now, cats are also interesting for another reason. They are a really nice demonstration of something else that's important to consider, when you think about evolution, and that's that traits don't evolve independently of each other. You have correlations among traits that really shape how species evolve. Now, Darwin noticed this, and he was kind of obsessed with white cats with different colored eyes because they're almost always deaf. And he hypothesized that there would be some sort of a link, obviously not really understanding that it was a genetic link, but a linkage between these traits, which we of course now know to be the case. And a huge amount of my research over the years has been thinking about how these trait linkages, which are actually driven by the genetic and developmental relationships among traits, how they shape evolution on large timescales. And so this kind of Saturn-looking plot on the side is actually just a really simple simulation I ran that shows how species could evolve if all traits were able to change independently. So if your finger length had nothing to do with your toe length, or your arms and legs were completely independently evolving. The red dots are what you would evolve, just give time and space, and you just get things dispersing through a morphospace, which is essentially just a theoretical plot of shapes that could evolve. But because if you just impose a little bit of linkages among traits, what you actually get is a distribution of those black dots. Now, this is mathematically kind of obvious, but helpful to think about, because actually what you can see here is that a whole lot of morphospace is never explored. And this is why, of course, a huge amount of things that we could imagine, like dragons, will never evolve because of those linkages between different traits. On the other hand, the flip side of that is that because there are limitations on what can evolve, the same forms actually tend to evolve over and over and over. And so this is actually a really good explanation for convergent evolution. Now, the plot on the other side is an earlier diagram that somebody made when they looked at the physical structure of shells and showed that all the shell shapes that we could think of as evolving couldn't possibly evolve because of these constraints on the relationships among different parts of it, but also just what's mathematically possible. And so it's really helpful for thinking about this. When you think about the evolution of life, it's not just that there's these external constraints on how things can evolve, but actually there's a lot of internal constraints on what can evolve. And we came up with this idea called the fly in the tube model. And essentially it's that there's this tube of space, which is dictated by development and genetics. But within that tube of space, you can get a lot of crazy evolution. Basically, everything that we study in evolution is what this fly is doing, zipping around in space, but in a relatively constrained space. But again, because that space is constrained, we tend to evolve the same things over and over again under the same conditions. So for example, these two skulls here, one is a thylacine, one is a wolf. Unless you're a mammologist, it is extremely hard to tell those skulls apart. There's a few key characteristics you can identify, but otherwise they look very similar. And one is a marsupial and one is a placental, but actually in terms of development, they basically develop very, very similarly and under similar ecological constraints or pressures evolve something very similar. So this is all part of kind of my work trying to understand why life looks the way it does, bringing these kinds of factors together with some of those extrinsic environmental factors. And one of the really important things I also consider is not just why life looks the way it does today, but how it does in the past. Because of course, the vast majority of species that have ever lived are extinct. And that also means the vast majority of diversity that has ever existed is extinct. And so we really want to take into account all this extinct diversity when we try to understand the processes that shape life on Earth today. Because without them, we would get things quite wrong. And I don't just mean if you think about the deep, deep, deep past, you wouldn't necessarily think about those dinosaurs up in the corner as ancestral birds, of course, they look quite different. But even if we went back just 10,000 years ago and asked a person living then to draw us a picture of a sloth, they would draw this, right? They wouldn't draw the sloths that we have here at the zoo. And so understanding that past variation really impacts how we understand the pressures and the factors that shaped how life evolved today. Okay, so a lot of my work over the past few decades anyway, has been really trying to capture this variation of life on Earth and then build complex models to explain how it has evolved. And so what we do in my lab is actually use 3D imaging to do this. So we go around the world, we visit collections around the world, we also dig up some of our own stuff and try to build a a good representation of the diversity of vertebrates in particular. And so that was one of my former postdocs, scanning some dinosaur skulls, and all the dots in this picture are some of the museum collections that we have visited over the years to try and build up this sample. And so we have, over 5,000 species. That's actually probably easily double that actually, in terms of what we actually have, but that's how much we've actually got through so far of our data. I'll come to why in a second. Now, once we have all this data, we have to figure out what to do with it. And you think that going around the world with a bunch of scanners is going to be the hard part, but actually that was the easy part. Because once you have all this data, you have to figure out how to compare things. And it's actually really, really hard to compare things that are very different. Now, the old way of doing this, and certainly when I was a graduate student, the normal thing to do would be to take some linear measurements. But linear measurements are a really poor explanation for the complexity and diversity of organismal form. You could also do things like use 3D coordinates for specific points, and that's what's shown in B here. But actually, if you want the exact same point on all of your different specimens, there's very, very few that you can capture. And what we actually want is a really nice, comparable high-resolution definition of this diversity. Now, across our entire data set of all vertebrates, we could only come up with about 12 points that we could find consistently across all of our skulls. And we can run an analysis of that. This is an analysis of about 1,500 vertebrate species, by far the biggest analysis that anybody has ever done. And to me, a little bit boring, because all it really tells you is that birds are weird. Yeah. Sure, that's great. You can do fun things like you can take skulls. This is a fox skull warped to what it would look like if it was a bird on the bottom. But it doesn't give you a lot of information about the real diversity of skull shapes. And so a lot of what we do in my lab is actually come up with novel methods for trying to capture complex phenomenon. And so we spend, you think I'm a paleontologist, spend all my time digging up dinosaurs. I actually spend nearly all of my time coding. And so this is a method that we came up with to really capture in high resolution the shape of skulls. And if any of you are ever interested in doing this kind of thing, we wrote a really tedious paper outlining all the things that will go wrong. But when it goes right, you end up with this really lovely depiction of skull diversity. We mainly look at skulls because they have lots of interesting features associated with ecology and development. But we can also look at all the different parts of the skull, and we can use that in combination with our understanding of the relationships of the tree of life, which is in the background there, to then reconstruct how this diversity evolved over hundreds of millions of years. Now, I say that as though it's really easy. It is really not very easy. It is certainly a lot harder than we thought it was when I wrote the grant to do that. And so a lot of our time more recently has been on figuring out how to make this much easier. And of course, the answer to that is AI. And so a lot of what our time has been in is developing new AI models to actually make that work a lot easier. And so I led the mammal part of that project. It took me about 7 years to collect the data that I'll show you in a few slides. One of my PhD students reran it with our new methods, and it took me 8 hours, which is fine. It did not break my heart at all. Yeah, it's just the 2000 teens completely wasted. So we've been developing a number of models trying to basically take these processes that used to take months or years to do, and we can now do them literally in seconds. So it's completely opened up a new frontier. And from my perspective, this is the absolutely most exciting time to be somebody who studies organismal diversity, because you can actually, for the first time, capture high resolution data for a huge diversity of species. Which is kind of ironic that I'm taking a bit of a step back for a few years, but I'm sure it'll be even faster when I come back to it. Okay, but once we have these data, we can do some really interesting things. And so I'm going to just give you a couple of examples of some of the work that we've been doing. Starting off with, a lot of people's favorite group, looking at the dinosaurs. And so we have, this is just a little image of some of the dinosaur and bird skulls in our data set. And with this new method, originally comparing dinosaurs and birds in a unified analysis would be basically impossible. You can kind of do a couple linear measurements and nothing else, because the change between dinosaurs and birds is so dramatic that they have very few points in common. And in fact, it's even really hard to identify individual bones in bird skulls, actually. But with our new methods, we can actually do this quite robustly. And so this picture up in the corner here is what we call a principal components analysis. It's a way of reducing really complex data into just a few dimensions that capture the variation. The silhouettes in the back of that are the rough shapes that each of the little quadrants represent. And the black dots are different bird species, and the color dots are different dinosaurs. Now, obviously, we have a lot more birds than dinosaurs in our data set, but hopefully you can see that the dinosaurs actually are spread quite broadly across the diagram. And that represents the fact that dinosaurs are actually incredibly diverse. Now, what we can also show up here in this other figure here is that where we find diversity in a skull is not even. It's actually extremely driven by the developmental origins of different parts of the skull. So some parts of the skull can evolve really, really rapidly, and other parts not so much. So things that are associated with the face, this tissue called the cranial neural crest, is extremely responsive to changes and can evolve really quickly. Other parts are much more conservative. And this is a really critical part of understanding, again, why life looks the way that it does. Not all things can change the same way, not because of development, and as we'll see shortly, also things respond differently to environmental change. Now we can also put all this together, our dinosaurs, our birds together, with our tree of their relationships, and reconstruct the pace of evolution, the speed at which things have changed. And one thing that was quite unexpected is that when you get to birds, so the colors here, the reds are really fast rates of evolution, the blues are slow rates of evolution. And hopefully you can see that for some of these plots, all of the slow rates tend to be in the birds, which are in the outer ring, and the inner ring are the dinosaurs. And we can show this a little bit better if we just compare. Now here, actually, do I have a laser pointer? I do, don't I? So up here in this plot, these over here are birds. These are non-avian theropods. That's a group of dinosaurs that birds evolved from. And over here, these are the other kinds of dinosaurs like your sauropods and your triceratops and things like that. And on the y-axis is the rate of evolution. And these are different parts of the skull. And in pretty much every case, the birds are slow. And that's surprising, of course, because birds are incredibly diverse. But actually, when you think about it, what birds and dinosaurs do quite differently is that dinosaurs use their skull, the hard parts of their skull, which is what we're measuring here, to have really diverse feeding types. So they chew a lot, they have lots of different jaw processing, they have lots of bony ornamentation like horns and things like that. They fight a lot more with their skulls. Birds, on the other hand, and no disrespect for the bird people here, but they're basically just a brain case with a beak stuck on it. You know, all that fun stuff they do with feathers. They don't do that with their skulls. And so what this really shows, although at first it was a bit surprising, is a change in the way that these animals are using their skulls. And that is reflected in the pace of their evolutionary change. So birds, it's a huge, huge downstep in the rate of evolutionary change when it comes to their skulls. They do all those things with their soft tissues. Now, when we look at things like salamanders and frogs, which actually has turned out to be my favorite group, we also find something quite surprising. Now, with amphibians, of course, they have this incredible diversity in how they develop. And what we found is that this life history is really what drives their speed of evolution. So in these plots over here, again, we're using the same kind of really high resolution description. This is again one of these morphospaces. And hopefully you can see the dots here, but the dots here all represent how they develop. So are they direct developers? Do they go through a metamorphic stage? Or are they paedomorphic in that they stay as a juvenile stage, you know, like your axolotls? And what we see is that they group based on color, not based on their relationships. And that means that the things that develop similarly actually look more similar to each other. Now, within salamanders, actually, how they develop changes quite frequently in their evolution. There's dozens of shifts in their developmental mode. And when we look at where the changes are in their phylogeny in terms of their speed of evolution, every time we see a change in how they develop, we see a change in how quickly they evolve. And in fact, when we compare the different modes of evolution, I'm not sure if that's very visible to you guys, but what we can see is direct developers are extremely slow evolving, whereas metamorphic and paedomorphic forms are really fast evolving. And so this has important implications, right, for how things actually will be able to respond to changes to their environment, to how quickly they can actually evolve. Now, we also actually looked at this in our frogs, and unexpectedly, we found the opposite. really metamorphic. Well, so all frogs are metamorphic, so it's not quite the same comparison as in salamanders. But one of the big differences in frogs is that what we're here calling a direct developer are those that have a larval stage that does not have an independent life mode. So it doesn't independently feed as a larvae. It's born, but just kind of hangs out, and then it metamorphoses and then it eats. A biphasic one are the ones where you have your free living tadpoles that are actually doing something. So you can think of direct developers as really lazy tadpoles, and these ones are active tadpoles. And it turns out that the ones that don't have this active larval stage evolve much faster than the ones that do. So that suggests that even having this functional pressure at that early life stage is actually really constraining the rate of evolution of these metamorphic or these biphasic frogs compared to the ones that don't need sort of two different kinds of feeding and two different kinds of food supply at different life stages. So again, this shows some factors that are going to be quite critical when we think about how species are going to be able to respond to change. And then finally, I'm just going to mention the stuff that took me 7 years to do. Since it took me 7 years, I should mention it. These are the mammals, my favorite group. And again, here's an analysis of their shape range using the same kind of data. This is again a morphospace. And what we see here actually, the colors here actually represent how many different species are kind of in the same area of morphospace. And one thing that's really interesting is that mammals are incredibly convergent. So every different group of mammals has something that kind of looks like a fox in it. So we see a huge amount of convergent evolution within mammals. And the only things that really are distinct are the whales, which are over here. Maybe that's not so much of a surprise. And also the rodents that are over here do something quite unusual with their faces. What was surprising, however, is that there are a lot of species that actually sit halfway between kind of normal mammals, as we can call them, and whales, which are decidedly not normal mammals. So for example, elephants, a lot of extinct forms, like these really unusual South American native ungulates that went extinct in the last million years or so, slightly less. A lot of them do something very similar as whales, where they're pulling back their nasals, probably to support a big proboscis. And so that's something that happens quite a lot too. So even the unusual things actually have a real shared pathway of convergence in becoming really unusual. Although nobody takes it to quite the extremes of some of these whales. Now, again, we can take all of that and put it together with our understanding of the tree of life for these different groups. So these are some of the mammals in our data set. This is their phylogenetic tree or their tree of relationships. And again, the red branches here are the ones where we see really, really fast evolution. And the blue ones are where we see slow evolution. So for example, we see really fast evolution persistently throughout whale evolution. Although there are some things that shift that, as I'll show you in a moment. And interestingly, groups like the rodents down here and the bats, which are, where are bats? Somewhere over here, here are bats. They actually don't evolve very quickly. They're incredibly speciose, right? 3/4 of mammal diversity is in these groups. But actually, they don't have a huge rate of evolution in terms of their anatomical evolution. They actually are very slow and steady in comparison to some of the other things that are out here. Now we can also take all this and combine this with some of the traits that we're interested in terms of their ecology and their development. So we can look at things like diet and locomotion, habitat. We also looked at developmental mode and other aspects. Now things like herbivores evolve fast, aquatic species consistently evolve fast. Habitat, other than aquatic, not necessarily so different. But the ones that I was really surprised by actually were things like precociality. So the species that are born at quite an advanced stage, the things that are in every nature documentary, running away from a cheetah, these things evolve quite fast. I think probably driven by this next aspect, which is social structure. Now, I actually ran all of these analyses during the pandemic in that lovely period, not lovely for obviously many reasons, but lovely from an academic perspective because it was before everybody started using Teams regularly, so you could actually were left alone quite a lot. And what I found actually was that social species evolve much, much faster than solitary species, which, when you are in isolation, feels about right. And then the last thing that was really interesting is that when we look at activity patterns, species that don't have a very fixed activity pattern, things that can really modulate when they're awake or asleep based on what's going on around them, actually have a much faster rate of evolution than things with a much more fixed activity pattern. So these are all things that what we'd like to do going forward is put them into models where we can start to predict with more resolution how species respond to environmental change. And to do that, what we're doing is building new models of new evolutionary models where we can directly input environmental data into the same models as we're putting these other traits into. So here's an example, just showing our birds here, but actually what I'm going to talk about are the whales, where we have our tree, we have our traits, we have our different skulls or whatever. We also have our climate data, climate information. So we looked at things like temperature change and precipitation. And because we have fossils in our data set, we can actually go back tens to hundreds of millions of years in our analyses. And what we can do is use the environmental change as a predictor variable for our evolutionary change. And we can see if that's better than, kind of a more standard model of evolution. And what we actually found in our analysis of our whales in particular, which I think is quite interesting, and here I have toothed whales showing here, is that different groups obviously respond differently to different variables. I don't show the baleen whales here, but baleen whales track productivity in the oceans. Maybe that's not so surprising if you've ever seen a baleen whale. They eat a lot, and basically all they care about is their food. So over the last, say, 25 million years, baleen whale evolution, the strongest predictor of how fast they're evolving is how much food there is around. Toothed whales, on the other hand, do not follow productivity. They follow temperature, but not just temperature. So we looked at not just the raw variables, we looked at the change in that variable through time, and we also looked at the rate of change in those variables through time. So the best predictor of toothed whale evolution over the last 25 million years is the rate of change in temperature through time. So I think I don't need to push this point any further, but I think you can see why this is actually really important for us to understand what species are responding to what kinds of variables. So this is going to be really critical for thinking about how they're going to respond going forward. And then, of course, the last thing that we are really interested in is extinction. As I started off in the beginning, one of the things I really work on a lot in the field part of my life is the last mass extinction. And with all these data sets, we really wanted to look at past mass extinctions and how different groups respond to them. So this is kind of an experimental piece coming forward. One of my postdocs had the idea to take these plots that we have where we look at time. See if it'll go forward. When we look at time on the x-axis here, you can see the meteor there is obviously the end Cretaceous mass extinction. I'm going to start off with mammals here, and we know that this was a really transformational moment for mammal evolution. So one of my postdocs was a violinist, and he had the idea to put these different plots to music. So for all the Kraftwerk fans in the audience, this is for you. So this is essentially the pitch is related to the pace of evolution over the last 70 million years or so. So this peak right here is right after the end Cretaceous mass extinction. Mammals take quite a hit, but then they of course bounce back. Quite good for us overall. This peak right here is actually related most likely to the evolution of whales. Bats also show up, and actually a lot of the major groups show up. But then this right here is sitting right around the mid-Eocene climatic optimum. We have another bump here in the mid-Miocene climatic optimum. Now, this model of evolution is something we call the attenuated model, where you have a big initial radiation, but you have later smaller peaks that are likely related to large-scale climate change, most likely. Now, because of issues with the dating of trees and things, it's not absolutely sure, but it certainly seems to match in many cases. Now, of course, not every group has such a volatile history. We have crocs. Crocs are pretty much just fine for most of the time. They're just kind of, hanging out. Still fine, If you're doing it right, why change? They're kind of like the cats of, the reptile world. There's a little bit of innovation happening in the Cretaceous. Actually, I think the Cretaceous is a fantastic time for crocodiles because of these guys right here. These are Deniscidians. They're my favorite crocodiles to find. They were small, they're herbivorous, they're terrestrial. They would have made great pets, I think. Of course, crocs take quite a hit in the Cretaceous mass extinction. It is, but they also have a bit of a boom when we have these climatic hot periods in the Cenozoic. And actually, crocs are actually diversifying now fairly well, especially in kind of Australasia. Just one more of these, I promise. Of course, extinction can be pretty bad for some groups. So here we have the dinosaurs coming through, and in the Jurassic, it's really when they find their moment, they're really diversifying. It's when you get all of your sauropods going in, showing up. The Cretaceous, obviously at the transition there's a bit of a little bit of an extinction, but they bounce back. They're really doing a lot of new innovations in the Cretaceous. This is when you're getting a lot of your big theropod evolution happening and your Triceratops and groups like that showing up. There's been a lot of talk about whether or not dinosaurs were on the decline prior to the mass extinction, but certainly from our data, there's no suggestion that that was the case. In fact, they really seem to be innovating in those final years of their pre-bird existence, or pre-bird domination existence. And then I'm not going to play the music for this because it is quite a cacophony, but as this plays, you can of course put all of these into the same scenario and look at how different groups are responding to these same events over time. And so here we have our crocodiles, we have our dinosaurs, turtles. You'll see some of the other groups that, although because this is based on our data, some of these groups like birds and mammals obviously have a Mesozoic record, but it's quite poor and not very diverse. But as we go through time, you can see the different groups really coming in, and some groups like the like the lizards and the snakes, as we'll see, are similar to the crocodiles, and they largely just kind of plod along at a fairly static rate. So they just have a kind of consistent rate of evolution. Other groups have much more volatile periods. So you see a lot of innovation in salamanders at various points, turtles as well. And then birds and mammals are the ones that are really showing this huge response in the wake of the end Cretaceous mass extinction until the modern day. But you can certainly see that some groups are really still innovating. We get more recent rises. And so I spent a lot of time looking at plots like this and looking at changes over time to different groups of organisms. And of course, anybody who spends a lot of time thinking about the diversity of life on Earth and the diversity of how that diversity changes through time can't help but always be very depressed by things like this, which I'm sure all of you have seen many times. And people look at these and pull different things out. I look at numbers like this, and even though, of course, we're talking about kind of population sizes and things like that here, I look at a number like this and I think, yeah, that was the end Permian mass extinction, or the Great Dying, when we hit about 94% extinction rates, obviously in species as opposed to populations. These numbers here, these look like the end Cretaceous, where 2/3 are going extinct. But of course, as you all know, we are the meteor in this instance, and there's no really secret or mystery to figure out what is happening. And so, increasingly, I think all of us, and I know all of you in this room can appreciate this, all of us who work on living species are increasingly wringing our hands over what we can do about this. And certainly, over this past week, I was at a conference, at a grant panel meeting in the EU, in Brussels for the EU. And a lot of us were talking about, we are reviewing grants for early career researchers, and we are talking a lot about what drives researchers. And somebody proposed that people are driven by two different things. They're either driven by pure curiosity, or they're driven by the desire to save the world. And I challenge this notion. I think you can be driven by both. I think curiosity about the world is what drives the passion to save it. And certainly that has been my experience. And as Matthew mentioned, I'm really about to shift from being essentially a pure researcher to somebody more involved directly with policy. And part of that is the fact that when I was making my career decisions in the 90s, I assumed that somebody would have fixed this by now. But here we are. And so certainly, I know all of us in this room are dedicated to making sure that we do what we can to prevent us really seeing out another mass extinction. Because again, when you look at mass extinctions through the past, you look at the winners, you look at the losers. The truth is everybody is a loser in a mass extinction. It was a squeaker for the birds and the mammals. It's shocking that any of us survived it at all. And while we look for rules, we look for predictor variables, we spend a lot of time and effort doing this. Fact is, when you're starting to hit those numbers, it gets very, very hard to predict what is going to happen. So I propose that we don't find out, and I'm sure you all agree with that. And so I'll leave you with that last thought with Charles Darwin, and happy to take any questions.
Thank you. That was absolutely brilliant. And as an organization which is basically all about a curiosity-driven desire to save the world. I think we would agree with you that doing both is not just possible but necessary. Am I allowed to say I prefer the science to the music?
They'll come back to you in the middle of the night, though. You'll want to hear it again.
I hope not. Questions? Ray? And we were relying on you to start.
You are, okay, thank you. Superb. Is your work able to practically give any guidance to conservation? And one thing I was thinking of towards the end of your talk was that I'm very interested in conserving evolutionary possibilities on Earth, which is difficult with tiny populations and so forth. It's part of a poster I've got at the back there, just a thing on orangs. I'm just wondering how we could possibly, as well as saving the actual species, keep the prospect of future evolution, because we're losing a lot of, in my mind, we're losing a lot of the possibilities with habitat loss. And
Yeah, so I mean, well, you just think about the basic equation of evolution, right? Variation. In order to have variation, you have to have healthy population numbers, as well as healthy environments and a variety of space, essentially, right, for species to move around into. And so I think that is a critical part of it, is having healthy ecosystems and healthy population sizes for species is a really critical part of it. I think the other part of this, though, in terms of what's useful for specific guidance on conservation is, when we think about the really big and really fantastic projects that people are doing, trying to predict where the greatest risk will be, where the greatest future risks may be. A lot of that is based on occurrences, because for the vast majority of species that have even been named, we actually don't know that much about what they do, right? So we always will need more observational data. But what we can do with analyses like the ones I showed you is then start to take our understanding of those traits and build them into those models, so we can better say not just from where species live, but what they do. How are they going to actually be impacted by change?
Amazing. Thank you. I think we have, great thing is we have lots of people watching online as well, and Amelia is going to read out one of their questions.
Hello, and thank you very much for your talk. That was unbelievable. So the question from online is, why do you think that crocodiles and salamanders are still diversifying so extremely in the current age compared to other species groups?
Yeah. Okay. So, well, two things to really clarify. One of that is I'm talking about the variety of forms and not the numbers of species. So other groups are definitely diversifying in terms of species, but in terms of the different forms and environments, part of it is where they are living, and in some of those that will be island driven, or it'll be the diversity of the different kind of microhabitats. In the case of salamanders, salamanders are also, I am not a herpetologist, so my passion for salamanders is fairly fresh. Anybody in here can disagree or can correct me. But salamanders are incredibly variable in how they develop and in their life history. And they certainly seem to be able to change in those ways quite quickly. And so I think that gives a lot of fodder, sort of going back to the cat and dog example, that diversity of development gives a lot of fodder for change. Now, there's also the aspect that having these kinds of complex life histories, I think, could be quite a constraint from conservation perspective, because you need sort of multiple environments to be preserved. But part of that is, a lot of it is kind of what parts of the world they're living in and how recently some of those areas have changed because of sea level rises. So in Australasia, for example.
Hello. I just wanted to know about the recent sort of news about bringing the woolly mammoth back and the saber-toothed tiger. What are your views? And obviously that's quite dangerous, but just what are your views on that?
So I've spent most of the past week actually arguing about this with various people. I would say my first view about it is that there are more efficient ways to do conservation. We are kind of in I'm sure you know this, we're in a period of global warming, so I'm not sure what the logic is in bringing back ice age mammals. So I would say that money, if the goal is conservation, and I don't know what the goals, the true goals are for the people who are funding that. If the primary goal is conservation, I think there are better ways of doing it. I think you could use that money more effectively to actually keep species that are going extinct now alive. There's an argument to be made for the development of really interesting new genetic engineering technologies that could potentially be harnessed to you know, de-extinct things that are at very, very low population sizes now. People have told me, people who are more embedded with that side of the field than I am, have said that is something that people are working on, artificial wombs and things like this. I don't think, from my personal opinion, I don't think we need woolly mammoths, and I don't think we need saber-toothed cats. I like elephants though, and I like tigers. So I'd love to see those not go extinct. I think there's, people suggest that it makes people excited about science. I think people can be excited about science for other reasons as well. I think it's also important to recognize that when people talk about de-extinction, they're not talking about de-extinction from the perspective that of bringing species back. They're talking about functionally de-extincting something. So there's something that kind of looks like it. They're not bringing back a saber-toothed cat. They're bringing back a cat that has longer canines. They're bringing back a slightly bigger dog. They're bringing back an elephant that has, slightly woolly hair at best. These are not the same species. Those things are never coming back. So I think, if there was a way to bring back things that have very recently gone extinct due to human causes, I would love to see a thylacine. But I would also prefer to not see the things that are still alive today go extinct, and that I think should be the primary goal of conservation efforts, personally.
And Colossal, the company which is doing this, I think in its own literature now has stopped saying it's trying to bring back the woolly mammoth and is talking about creating a winter optimized elephant.
Yeah.
So, I think we have a question in the middle and another question online.
Given the relative incompleteness of the fossil record, what potential do you think there is for new fossils potentially changing some of the conclusion? Is that likely, or have you got a sort of wide, rigid enough data set that things are unlikely to change from now?
Oh no, I mean, we have still barely scratched the surface of the fossil record. In particular, the fossil record is heavily biased towards certain regions and times. So North America, Europe to some extent, China, these have really excellent fossil records. One of the things I tried to do about a decade ago is look at the latitudinal diversity gradient. We have more species in the tropics and in the poles. Try to look at how that changes through periods of climate change. So if we go back to the Cretaceous, which was significantly warmer than today, even if we go back to the Miocene, which was about 4 degrees warmer than today, what does that latitudinal gradient look like? And that project, we could reconstruct it for the Northern Hemisphere, and it actually shallows out quite a lot during warm periods. But we could not reconstruct it for the Southern Hemisphere with any statistical significance because the fossil record is extremely poor for a lot of the Southern Hemisphere. And so that's why most of my field work has been focused on those areas, because anything we add there is really very new. And certainly a lot of the stuff that we've discussed that we've discovered in places like India and Argentina and Madagascar has really rewritten the story for a lot of groups. So I think the patterns may well change to some extent, but with new discoveries, maybe the larger scale patterns less so, but I think we have a huge amount left to learn.
Thank you. And our last question goes online.
So where do you see the biggest opportunities for discovery in evolutionary biology in the next decade?
Oh, okay. Well, I think, as I said, it's a really exciting time to be somebody who works on phenotype, because we're really, with imaging, just getting the ability to build these huge databases and actually analyze huge databases. That has just, exploded in the last few years. What I'm really excited about is actually the ability to link genomes to phenomes properly, because we see all these papers where people have these full genomes and then they just look at body size. Well, phenotype is more interesting than just body size, right? And so I'm really excited about the ability to link genomes and phenomes together with real depth, and then use that to really, really understand what are the factors that shape how species change. And then hopefully linking all of that, obviously, with broader scale efforts to predict and hopefully conserve species. That's my pure evolutionary biology answer. What I think is the most important thing for the next 10 years, though, is actually to stop the human-caused extinction.
Thank you. I have to say, I wish my daughters had been here to listen to it, because I think it would have guaranteed they would become scientists. It's such a brilliant talk. Thank you very much.
Indeed. Thank you very much, Professor Goswami. You quite agree. We all were rebelling with you, I think, in evolutionary biology there. It was an incredible talk. Thank you. I now have the exciting task of handing out the prizes to both the poster winner and also the prize draw winner for the bingo cards. So I think probably a little rumble or something we need, but bingo card prize goes to Ray Heaton for the month. I'd like to now. And the second, it's for your cup of tea, for your bingo card. The second prize for the poster winner goes to Kelly Sambuki. Kelly, do you want to collect that afterwards? And the first prize poster goes to Kat Terrace for your vouchers. May I ask, when you get a moment, if you would fill in your feedback survey, either through the QR code on the programme or you'll see it in the link for those who are online. We'd really appreciate it. As Matthew said, this is our conference for you, for your event. So the more feedback we have, the better to improve it for next year. And may I thank you all for being part of this wonderful community of fellows. We just heard that social species evolve faster. So we should all learn from that. And indeed we are, aren't we? What an incredible day today, learning from so many experts and specialists in their field. And to that end, could I also ask you to think about if you'd spread the word about fellowship to those that you think would also be enjoy joining our community and being part of the ZSL Fellowship. If you've anyone to suggest, do talk to our team in green or make the suggestion direct to them. And just to that point on our team in green, could I ask for a quick round of applause to Lindsay, our fellowship manager, who has put all this together, Katie, who's delivered the event with real aplomb, and all of our fellows and supporters and volunteers here today who've made it possible. And before we gather for our final drinks and networking, I'd like to welcome Professor Sir Jim Smith back to the stage once more for our closing remarks.
Thank you, Pippa. I'll be very brief. Anjali, thanks for a terrific talk. It warmed the hearts of this old Evo Devo person enormously. And I have to tell you that I gave a talk to the Francis Crick Institute about a week and a half ago, where somebody asked me the very same question about, colossal biosciences. And I had to say that I gave a very similar answer, but because I knew I was among friends, I was able to be rather more robust in my language. If we had $10 billion, I think we could do a lot more than they are doing. So we agree. Thank you. I think it's been a fantastic day, and really my task is just to say thank you to everybody. First of all, actually to you, Pippa, for emceeing so well, and thank you for thanking our team so well, Lindsay, Katie, and also Harriet Makara. Lindsay isn't here today, I think, because she has COVID. So we wish her well, and I think let's wish Lindsay well. I'm grateful to our speakers, Louise, Javier, to our fellows who spoke, Patrick, Sarah, and Imani. I think you'll get a link to listen to Richard Kindlin. He'll be terribly disappointed you weren't able to hear him. But I know Richard, and I know it'll be a brilliant talk. So when you get the link, listen to it. It'll be worth it. Thank you to the fellows who volunteered to be ambassadors and help the day run so smoothly. We had a little fellowship working group from the council who were Athena Marquez and also Karen Lawrence. Karen's here. So thank you to you for your work. And really, the only other thing is, as Pippa said, this is your meeting, as Matthew said, it's your meeting, it's your society. We want to make your society work for you. We want this meeting to work for you as well as it can. So as Matthew said, we want to hear what went well, and we even want to hear what went badly. So things that go well, tell me. Things that went badly, tell Matthew. It's an old joke, Matthew, but it usually gets a laugh. So that's really all I have to say. I just want to thank you all again so much for coming. Thank you for making the day work so well. To thank the people who ran the sessions where I particularly learned about the library and the books in the library, learned about photography, other people learned about Drosophila and so on. Thanks to everybody for that. I think that was fun and worked well as well. And so now, as a fellowship, we are going to join together in fellowship and have a drink. So thanks everybody, and see you next year.